It's now time to look at the future and the organisations that have been honoured in this year's round of grants. As Ian alluded to, $8 million in total to 4 fantastic, I won't say organisations, I'll say projects, because as will become clear, some of these projects involve multiple organisations coming together. We're going to meet a representative of each of them, chat a little bit about the specific work and more generally this field in which they are involved. So on behalf of from Peter Mack and WeHi, he's a very complicated guy, it'll become apparent soon. Tony Pappenfuss is here. Give Tony a big round of applause. Come on forward, Tony. From the Centenary Institute, Philip Hogg. From the Institute for Molecular Biology, Jenny Stowe. And from the Alfred Hospital in Melbourne, Professor Andrew Spencer. This is our panel this evening. I'll get them each to quickly explain who they are, what it is the work they're doing that has been recently acknowledged by the Foundation. We'll then drill down. Starting with you, Tony. Peter Mack and WeHi, all part of the VCCC. What's the VCCC and what's this grant you've received for?
The VCCC is the Victorian Comprehensive Cancer Centre, and I'm the head of computational biology at both the Peter Mack and also the Walter and Eliza Hall Institute. The funding that we've received is for equipment that will help us to investigate tumour heterogeneity. So this is not just looking at cancers as one cell gone wrong, but as a whole population of cells, or more appropriately, an ecosystem of cells or different species that are genetically distinct and may respond to therapies in different ways, as well as immune cells that are infiltrating the tumour and stroma and other tissues that are supporting the cancer.
To paraphrase Tom Cruise, you had me at computational biology. You're a bit of a maths nerd, aren't you, Tony, like me?
I am a maths nerd.
What you're looking at here is, from what I understand in a very simple sense, if I have a certain cancer, all my cancer cells aren't necessarily the same. You and I might have the same cancer. Our cancer cells can also be different. Is that what tumour heterogeneity is about?
So not only might your cancer be genetically different from mine, different parts of my cancer can be different. They can have different mutations and respond to therapies in different ways.
And when you start to roll out those number of differences times this, times that, things become remarkably complicated, don't they, in terms of trying to find a fix?
That's right. And it's helpful to think about these things as a whole, as a population or as an ecosystem, as I said before.
And so what's the essence of the program? What are you aiming to do with this money? What's it particularly focused on in terms of tumour heterogeneity?
So we're applying this technology to a number of different types of cancer and investigating different aspects of their evolution and their heterogeneity. So this includes how cells are genetically different from each other, how they are, so genetically means mutations in the DNA, how they are epigenetically different from each other. So these are changes in marks that are on the DNA, and how immune cells infiltrate into the cancers in different ways.
It was at this function last year, I was chatting to Roger Riddell on a different topic, and he reflected on his own career and he said if he went back and started again now, the one thing he'd do differently is he would do more maths. And I obviously pushed the maths barrow, but it is this age of data. There's no shortage of numbers and zeros and ones we're sucking at. It's turning that out of noise and into information is the real challenge.
It's not the only challenge,
yeah.
But it's a big one. So I am the data nerd on this grant, and I'm very pleased to be that, because it's going to generate exciting data that we're going to be challenged with, and it's going to provide deep insights into what's going on in cancer.
Excellent. A round of applause, please, for Tony and the team. Great stuff. Now, Philip, you're at the Centenary Institute. You wear many hats, but in terms of this grant, Centenary Institute, explain the Institute to the audience and this particular work on sort of tumour metabolism, the diet of tumours. Talk us
Okay,
through.
so I lead the ACRF Centenary Cancer Research Centre at the Centenary Institute, which is an independent medical research institute on the University of Sydney campus. And this grant is about how tumours metabolise nutrients. So they do this in a fundamentally different way than our healthy cells do. So the main nutrient we get from our diet is the sugar, the glucose. And our healthy cells use this glucose mostly to make energy. And what they don't need, they store away as fat or glycogen. Whereas a tumour cell is really not so fussed about making energy. What they want to do is make new tumour cells. So they take the nutrients in our diet, particularly the glucose or the sugar, and they metabolize it in a way that makes new components of tumour cells. And so this fundamental shift in how they metabolize the glucose in our diet gives us an opportunity to target that aspect of tumour growth. I think the big advantage of it is it seems to be a fundamental property of most solid tumours. So if you can find a way to effectively target how they metabolize their sugar, you may have something that could be a good therapeutic.
It's my understanding that the whole concept of the diet of the tumour is not new. It actually goes back a long way. When did people first start considering this metabolism of nutrients?
Okay, so the original discovery is a guy called Otto Warburg. So Otto was a German clinician, and in 1930 he discovered that when he took some tumour cells and cultured them in a test tube, they made acid. But as the healthy cells didn't make acid. So he was clever enough to realise that the reason they were making acid was because they were metabolising their sugar in a different way. And I suppose that was a seminal observation, but it really lay dormant for 50 to 60 years, mainly because we didn't have the tools to really understand at a molecular level how the tumour cells were doing this or why they were doing it.
I was going to say, that's almost 100 years ago. Did they also, around that time, we start to understand genetics, DNA, does that sort of not distract, but I presume a lot of focus went in there too, because that's almost a century since he made that fundamental breakthrough. Why is it so hot as a concept now?
Okay, so there's a few reasons, but one of the main ones, really the last 30 years of cancer research has been dominated by the genetics of cancer. And we've made phenomenal discoveries and breakthroughs in that area. We understand it is a genetic disease. And the issue is that most tumours, they have lots and lots of mutations, 50 plus mutations in tumours. And the challenge is, and so what we're knowing now is that we can take out one of these mutated proteins, fine, but the tumour is so adept at finding ways around these specific functional blocks is that it's a challenge to take out every cell in your tumour, because there's going to be tumour cells there that just can find a way around it. And that realisation in the last 5 years probably that has led people to look at this, look at sort of some fundamental aspects of tumours that applies to all the tumour cells. And metabolism is one of those areas.
You're focusing, you say this could apply to a vast range, but there's 3 particularly nasty cancers that a lot of your research at the moment will home in on?
Yeah, so there are some tumours that this metabolic switch are particularly relevant. Tumours of the brain, a glioblastoma, this is your most malignant and most common brain tumour. The pancreas is another one. Ovarian cancer is another one, and liver cancer. And particularly some of those cancers, like ovarian and liver, there's a strong association with the diet. So in ovarian cancer, about 40% of it is attributed to obesity. In a liver cancer, it's about 20 to 25%. So in addition to these cancers really, they really undergo this metabolic shift, there's also an association with our diet that we really don't understand. Why is obesity associated with 40% of ovarian cancers? So if we can understand that link, this will lead to some good stuff.
Your grants for $2.5 million. You're the only person on the panel who was at the governor's function as well. I chatted with you last week, and I saw you receive the plaque that has an imprint of a $2.5 million cheque on it. From what I understand, you actually took it home and had a bit of fun.
Yeah, the boys thought it was for us. So,
Santa would have got a few pretty impressive letters going, actually, upgrade that PS4, buy me a small island in the Bahamas.
Yes, I had to set them straight, and they went, oh, okay. No.
Give Philip a big round of applause for the Centenary Institute grant. It's really exciting stuff. Jenny Stowes from the IMB, the Institute of Molecular Bioscience. Tell us a bit about the IMB and this Cancer Ultrastructure and Function Facility that the grant, I think I might have even got that right, that the grant is for. Tell us about the IMB and this specific grant.
Thanks, Adam. IMB is a multidisciplinary research institute, which means we have people working across many different areas on cancer, from chemistry to biology to development and even maths, which is very important. And so all of these fields come together to give us a really multidisciplinary take on what's going on in cancer.
Tell us about the Ultrastructure and Function Facility. This specific grant, what's it aiming at?
So we're very excited about this money because this funding is going to buy 3 of the latest microscopes that have been developed in the world. And these, for the first time, will let us watch tumour cells in real time, behaving as they do over several days. And so we can put a drug on the tumour cells and watch over several days what happens to that tumour cell, to see whether it dies or divides or moves or doesn't move. So this will really speed up drug discovery. It will also help us dissect the effects of gene mutations or metabolites on cells that are in tumours. It will also allow us to reconstruct tumours in 3 dimensions so that we can look at different parts of the tumour and see what they're all doing individually. And this will really help us design individual treatments for individual types of cancers and even individual patients that can much better target the cancer itself.
Correct me if I'm wrong, it's my limited understanding in terms of the 2 different parts of, let's say, a cancerous cell, the genetics, what is it made of versus how does it grow, change, evolve over time? We know a lot more about the what is it made of part than we do about the real time, but what does that become, how does that change, which must be a very big component of understanding how to deal with it.
Absolutely, Adam. So you're right, we're learning more and more about genotypes, the genes in tumours. We need to learn about phenotypes and what those genes make the cells do. And what they do ultimately is behave as part of a community of cells and also with the matrices and tissues around them that they respond to very strongly. And so looking at them in situ, in the context of a tumour, is really important to understanding exactly how to target them with drugs and how to control tumours.
And so this $2.3 million is going towards buying some pretty impressive toys. Talk us through the specs on some of these. You've been to the States and you've been inspecting one of them operating now?
Yes, so we've been going over to the States to use one of these microscopes that was built by the 2014 Nobel Prize winner, Eric Betzig. And Eric is working at a new field that's grown up as part of this whole imaging field, and that is the intersection of physics and maths with biology and computing. Eric really sits in that space, and for many years he couldn't get a proper job because he really didn't fit into any of the pigeonholes. So the microscope he got the Nobel Prize for, he built on the living room floor in his house. And that quickly morphed into some very high-duty, heavy-duty microscopes when he did get a proper job, which is now. And he's building microscopes faster than we can use them, and these are the really cutting-edge instruments that will put Australia on the map along with the rest of the world in being able to look at tumour cells. So we're very excited about this.
We're going to have one of them at the IMB in Queensland. Give Jenny a big round of applause. That's really exciting, Jenny. And Professor Andrew Spencer is from the Alfred Hospital in Melbourne. Our first 3, while quite different areas, but the first 3 projects all in some way having common understanding cells and what they're made of and what they do and how they work. Andrew's work is quite different. We're talking blood cancer and therapeutics. Blood cancers are interesting, Andrew, because I think when you say cancer to a lot of people, they instantly think of cancer of an organ. Lung cancer, skin cancer. Blood cancers are a big part of the cancer family. Are they as understood, as concentrated on as they should be? Loaded question to you.
Sure is. So there's a fundamental difference between blood or haematological cancers and solid cancers, in that haematological cancers, by definition, when you're diagnosed, are already widely disseminated. They're everywhere. So there's no role for a surgeon, for example, in the management of blood cancers. The other major factor is that because they largely involve the bone marrow, by definition, they have a huge impact on your ability to form normal blood cells, and they can be therefore very immunosuppressive. So the diseases themselves have quite an adverse impact on the well-being of the patient's immunity, which can then be further impacted on by the therapies that are used. So the treatment paradigm is very different in blood cancers and solid tumours. And they can't be prevented. There's no screening program for leukemia. You know, you can't have a mammogram to pick up early leukemia. If you've got it, you've got it.
Are there many ways in which all blood cancers are fundamentally the same, or are they incredibly varied, or almost a bit of both?
No, they're incredibly, the word is heterogeneous, and that was used before, tumour heterogeneity. And the two diseases that we have a particular interest in, for example, that the ACRF funding will help us to work on more closely, acute myeloid leukemia and multiple myeloma, could not be more different, in that acute myeloid leukemia genetically is a fairly simple disease. So maybe one or two mutations will cause that. Conversely, multiple myeloma is incredibly complicated genetically. It's completely messed up. So they're very, very different at a genetic level. And I think that's reflected across all the different blood cancers. They're all very, very different from each other.
Ian mentioned in a press release about this, he said that what he's hoping will happen here with this national centre is a virtuous cycle of drug discovery, validation, personalised molecular monitoring, improvement of treatment combinations, a potential flagship success on sort of international standard. What's he talking about when he talks about that virtuous cycle?
So my colleague Andrew, we're both called Andrew, makes it easy. We are clinicians, so we spend most of our time looking after patients, but we also run a very large number of clinical trials. So we normally have 70 or so trials opened at any one time. And a significant number of those trials are what we call investigator-initiated trials. So these are studies which are based on discoveries we've made in our laboratories, taken into the clinic, and we design the study, we own the data. It's not a pharmaceutical company trial. And one of the things we're very good at is procuring very comprehensive samples from the patient during their journey on these trials. So entry in the trial, when they respond, if unfortunately they relapse. We've got all sorts of samples we collect. And so there are immediately a lot of questions you can ask. So why do some people respond and others don't? Why do some people relapse, and what's the mechanism of relapse? And we can then take that back into our labs. And so the resources provided by the ACRF we can bring to bear immediately in asking these questions, because we already have large numbers of archival specimens from patients treated with novel therapeutics. And I think this will really change how we manage these particular blood cancers in the short term.
Give Andrew a big round of applause. Really exciting time. Let me ask you quickly in closing, going to the line, one question each. Tony, I mentioned you're at Peter Mac and WEHI. The VCCC, the collaboration that's coming together there is amazing. Can you talk to us quickly about the strength of that collaboration, and also the fact that basically all the different institutions that make up the VCCC have received significant ACRF funding at some time or another?
So there's VCCC is sort of two things. It's a building, which is a fantastic place to live and work, and it's 10 organisations working together in an alliance. And I actually sit in two of them. At the heart of it are a number of hospitals which provide us with access to patient samples. It's the home to a very large number of really talented researchers and clinicians. So it really operates in a way that's remarkable and a joy to be part of. The ACRF funding is really essential in helping us generate the data that we're going to use to help make sense of how cancer evolves and how it's heterogeneous, and what we need to do to treat it better.
Philip, Doug mentioned, sometimes what drives us is the failures, and dealing with those. You've as I said, you've been in this game for a while. Do the failures drive as much as the successes? What is it that keeps you going back? And in particular, Doug mentioned someone in his lab who had a breakthrough after just a massive breakthrough after just a few months of being there. Is that exciting, or does that piss you off? That some precocious little punk just walks in one day, throws a dart at a board, and bang.
Yeah, there's no doubt, being in the right place in the right time is important. But the job itself is about discovery. I mean, it really is discovery in its purest form. We're trying to understand how the body works and why it goes wrong. And so you don't make the breakthroughs very often. You can struggle for years, but when it happens, it's really something. It's a real adrenaline rush. That's why I think scientists, researchers are sometimes considered a little nerdy, because that sort of adrenaline rush you get from those discoveries, you sort of don't often get from the sort of social interactions.
Very diplomatically said. Eh? But yeah, that moment must be, when we those of us outside tend to sort of, probably simplify it and patronise it slightly, but those moments must be amazing.
Oh, they're really, they're terrific. I mean, you all of a sudden you realise something that no one knew before, and that's enormously rewarding. Often in research, though, it's the big surprises. You think, well, you're going in thinking, okay, we're going to get this result. This is my idea. I know how this works, and you often get the opposite result. And you often learn more from the opposite result than the one you expected. So it's a very humbling experience. There's no right or wrong way to do it. We all do it differently. There's a real art to the game, and that's the challenge and the beauty of it as well.
Jenny, can I ask you that now with this idea of the genetics now versus the growth and the development, so there's obviously a complexity there that we understand now that maybe we didn't understand 5 or 10 years ago. Are we close to the end of unravelling all that, or are we just stepping off into something that's going to get more and more confusing, or do we even know?
We're never at the end, so we'll never know everything about cancer cells, but I have to say technology of many types is advancing so rapidly at the moment, which is why ACRF is so important at the moment, because technology is allowing us to take big leaps forward in understanding. And so the big leaps forward in understanding now, many of them are coming at the interface of understanding how cancer cells operate in the context of whole tumours and in the body. And that offers us new opportunities to stop cancer, to treat cancer, and to target cancer with either new or existing drugs. So there are leaps forward, and it's really technology driven at the moment. Technology is giving us opportunities that didn't exist 3 years ago, let alone 5 years ago or 10 years ago. So it's an accelerating process, and I think it's really starting to show effects in the clinic for individual people.
And Andrew, you mentioned something towards the end of what you said in my first question that's really exciting about your work, that some of this stuff is quite ambitious and long-term planning, but you said that what you're doing is pretty much coming online now. Tell us a bit more about that. That must be exciting.
Yeah, it is, because as I said, the trials that we do, that we really want to bring this technology to bear on are trials that we've designed based on our own work preclinically. So this is work which wouldn't happen if it was up to the pharmaceutical industry to do it, because they may not be the financial imperative. But we look at interesting compounds that may have activity in these blood cancers, and really it generates not only very interesting clinical data, but questions about why we're seeing certain responses. So we see exceptional responses in some people, which are very prolonged. And so investigating why has that person got such an exceptional response is just as important, for example, as to working out why someone didn't respond them when they were supposed to. And as been alluded to, it's all boiling down to bringing to bear these high-cost technologies which tell us about genetics, epigenetics, so we can actually start investigating these questions immediately, because we're seeing these patients every day. We're clinicians treating these patients on our trials. So it'll be basically immediate, the benefit.
I hope we've managed to capture in that small snapshot just the breadth of these programs, the excitement of what's taking place in these labs, and I'm proud to say all around Australia. Please, a big round of applause for Tony Pappenfuss, Philip Hogg, Jenny Stowe and Andrew Spencer. Thank you so much to our panel.